Our next presenter is Jared McAuliffe. He's majoring in physics. He's mentored by Dr. Thaya Paramanathan in the Department of Physics, Photonics, and Optical Engineering. Jared's presentation is titled Exploring the Binding of Anti-Cancer Drug BI 3802 with DNA Using Optical Tweezers.
Hi everybody, happy Friday. So exploring, So I'm going to be talking to you about some exciting research here today. We explored the binding of anti-cancer drug using optical tweezers. So in the single molecule biophysics lab, we use optical tweezers to trap single DNA molecules and trap them in the presence of various concentrations of anti-cancer drugs in order to observe their interactions. So I just want to point out to you here on the left side and the right side here is the structure of DNA. It's got the double helix, and that's connected by base pairs. You can kind of think of it as a twisted ladder. So that's going to come into play later in the talk. So you might be asking, what are optical tweezers? So optical tweezers, much like household tweezers, you can trap a tick and pull it off your dog. Optical tweezers use a highly focused laser beam in order to trap biological materials on the micron level. So if you look here, we have the laser. It's split into two. That red line represents the laser path. It's split into two, comes back together through these two objectives here, which focuses the beam inside a flow cell. So a flow cell, I have one here. It's a custom plexiglass chamber where all our experiments take place. So this is a cartoon representation of inside a flow cell. This black triangle here is a micropipette tip. The red dot is our laser dot, and that circle around the red dot is a bead that we have just trapped with the laser dot. So that's our first step in trapping a single DNA molecule. Then we're going to take that pipette tip, bring it over, enact the pressure on that first bead, take it out of the trap, and wait for another bead to be caught. So now that we have two beads trapped, we're going to start the flow of our DNA. And one end of our DNA is going to be chemically attached to the bead in the trap. Then we're going to move that micropipette tip. That way the other end of the DNA is chemically attached to the other bead on the micropipette tip. So now we've successfully caught a single DNA molecule, and we're ready to stretch it. So when you stretch a single DNA molecule, you obtain a force versus extension graph, as shown here. So originally when we stretch the DNA, if you imagine this as the DNA, it's slack and it's stretched to be taut, so not much force is exerted. That's that red section down there. Then once the DNA becomes taut, it begins to act as an elastic, so a lot of force is required for not much extension. That's that green section there. And then around 65 piconewtons right there, which is a force measurement, it's believed that the DNA base pairs begin to open up, like the DNA base pairs we were talking about in the structure earlier, and that's known as DNA melting. So at the end of that transition, you'll have two single strands of DNA, which once again act like an elastic, as shown in that purple section there. Okay, so this figure on the left here is a DNA structure, and the DNA structure bound by intercalators that are highlighted in red. Intercalators are a common form of chemotherapy drug. They're known to stack in between the base pairs of DNA, which in turn lengthens the DNA and as well as strengthens the DNA's double-stranded structure. So this graph is a graph showing DNA stretched in the presence of various concentrations of a common intercalating drug. So what you can see here is as the concentration of the drug is increased, the length of the DNA is increased. And this pink arrow shows here that as the concentration of the drug is increased, the force required to reach that melting transition, where the base pairs open up, is also increased. So since more force is required to open up the base pairs, that means the DNA structure is strengthened. So this is the chemical structure of the drug I'm studying, BI 3802. It targets a transcription repressor in order to stop the replication of cancerous cells. The figure on the right here, we have circled this flat planar section that makes us believe that the drug can intercalate to DNA. and stacked between those base pairs like we've been talking about earlier. So BI3802 is dissolved in DMSO. DMSO has not been previously studied in our lab. So first we had to test its effects on the trapping strength of our laser. We tested 5 different concentrations of DMSO. On this left graph here, you'll see the 1, the 2, and the 5 microliter DMSO additions into 2 milliliters of buffer, where no effect was observed. And the graph on the right shows the 10 and the 20 microliter DMSO additions into 2 milliliters of buffer, where an effect was observed. You can see the trapping strength is actually lowered. We also had to test if DMSO had any effects on the stretching of DNA. So once again, the graph on the left there shows the 2 and the 5 microliter additions, where no effect was observed. And again on the right, the 10 and the 20 microliter additions, where an effect was observed. So since, based on these tests, we realized that as long as we stay under 10 microliters of DMSO added into 2 milliliters of buffer, our experiments won't be affected by DMSO. So then we can move on to test our drug. So far we have gathered data on 3 different concentrations of BI3802. This graph on the left here shows the 1 micromolar BI3802 concentration with the blue curve. In the middle with the green curve is the 10 micromolar BI3802 concentration. And on the right with the red curve is the 50 micromolar BI3802 concentration. You can see the black curve in all 3 graphs. That represents DNA stretched in the presence of no BI3802. There's a trend observed on these 3 graphs where the melting force, the force required to reach the melting transition is lowered. So based on that data, the force required to reach the melting transition is lowered, which isn't actually common with intercalators. So we believe that this drug is binding in a different way than an intercalator. So we plan on collecting more data on BI3802 in the 50 micromolar and additionally 100 micromolar range. Then we can analyze that data, take that analyzed data, fit it to binding models, and based on those binding models, we can get binding characteristics like how quick it binds to DNA, the structural change of DNA after it's bound, and where on the DNA it likes to bind to. And based on these binding characteristics, we can hope to help develop better anti-cancer drugs. So I just want to thank my mentor, Dr. Thaya Paramathan, for all the help he's given me on this, and the Adrian Tinsley program for a great summer. I've learned so much over the summer, and then some acknowledgments here as well. I'm happy to field any questions. Yes?
So if BI3802,
Yep.
so if it lowers the melting transition, that means it's easier for the DNA to break apart?
It's easier for the base pairs to open up,
Yeah.
correct? Yeah.
So how come, do you know why it's used as a cancer drug if it makes it easier instead of harder?
It's actually used as a cancer drug in much lower concentrations. So they probably won't even see any of the effects on DNA that we see here. Yeah. Next now.
Why is it like a good thing for the DNA, like for it to like break apart at lower parts? Like what advantage does that give you in a lab, like when treating cancer?
So a lot of the, like we said, a lot of the chemotherapy drugs are intercalators. So they'll actually strengthen the DNA structure as opposed to this one. So it should make it harder for them to open up. That way, because cancer is the constant replication of cells. So it targets a specific area, usually a specific gene on the DNA. And it's either stops or slows that replication. So that's how it works with cancer.
So it's not a good thing that this.
Well, this just binds with DNA in a different way. We haven't collected enough data to see how it binds yet. But so we can see that it doesn't bind as an intercalator. Yeah. Yes?
How are you measuring force?
We measure force as a, it's actually a function of, I should know this too. Diffraction, yes. So the laser is diffracted from both sides, and we can measure that diffracted laser. And so by the movement in the bead, by the tugging of the DNA. We can measure that force by the diffracted laser beams.
I just want you all to know that when Jared didn't quite get that word, all Dr. Paramanathan did was go like this, and he said diffracted.